Team Centauri's goal is to model the molecular evolution of fast-mutating nucleotide sequences such as influenza.
Our project is targeted towards research and education.
Researchers will be able to see possible genetic outcomes of the viruses over a number of generations for uses such as phylogenetic analysis and possibly developing vaccines.
It can also be used for teaching students about the evolution of viruses and what they look like. Currently, researchers have done this in the lab with live viruses by changing the environment to see how they evolve. This has become controversial because if they somehow get out,
Well, let's just say, wouldn't it be fun?
it would be far safer to model or project these viruses' evolution in a simulated environment. Biology students can learn about amino acids and how different mutations occur in nature.
Students in computer science and similar classes can learn how bioinformatics and phylogenetics can be applied to these fields. Essentially, in the end, we intend to have a whole framework that will be open for research and students all over the world. This is the main workbench. For this demo, we're going to create a new project and run a ViraComp simulation. I'm going to name my project Test, and we're going to go ahead and create a local database, which we'll name TestDB. Finish. Now, since we know that this database and project are completely new, we just created them, we know that there's nothing in it, so we need to add a sample set now. I have a GenBank sequence already ready that has rhinovirus B nucleotide sequences already in it. And so I'm going to just call the dataset Rhino. And now it has been added to our project. There's all the information about the current sequences in table Rhino. Before you can run a ViraComp simulation, you have to run a pool alignment. We're just going to use the default values, which is a global alignment. Once you've run an alignment, you can then run a ViraComp simulation. ViraComp allows you to combine your simulated sequences with the parent pool, meaning that if I want to create 10 new sequences based off of the original pool, I can also include the original sequences in the same file. That way it's easier when I'm exporting the file later for third-party or custom analysis of the information. That way you don't have to export two different files. With ViraComp you can also run a alignment of the simulation. This way you can just align a simulation before you export it, so you don't have to do it in third-party software later. And we'll use the same settings we used previously. Once finished, you could export your simulations, which you can see here we have two. This is the simulation we ran, and then this is the alignment of the simulation that we ran. So I want to export the alignment to my desktop, and there's our alignment. ViraComp also allows de novo assembly tests to be run. You can also search for particular genes or amino acids found in the entire project. The UI can be customized to any particular way that you like it, as so. You can view project information, and then you can also export logs in case you're having problems. That way we can read the logs later. You can also export the original sequence pools, alignments, or assemblies. Say if I wanted to export our original Rhino pool, I would just go export sequence pool, and then I would export it to my desktop or wherever I want to save it, and it exported in FASTA format. I can import new sequences to this project. Say I can rename this one Rhino 2, and Rhino 2 now exists within this project. Or I can also import entire folders filled with sequence files in GenBank or FASTA format.
We call it ViraComp, modeling our lives by modeling the future.